Okay. Hi, so we're going to get started. So today we have Jason Stein from the Department of Genetics and the Neuroscience Center at the University of North Carolina at Chapel Hill. He explores how variations in the genome change structure and development of the brain. And he'll be talking today about some published work from his recent publication in Science and bioRxiv preprint on chromatin accessibility in neural differentiation. So with that being said, I'm going to turn things over to Jason. Thank you so much for joining us today.
Great, thank you so much, Shira. Thanks for inviting me for this talk. I hope everybody out there in internet land is safe and healthy and staying sane. Hopefully this talk for an hour can provide a little bit of distraction on some science. So I'm going to tell you about two projects, like Shira just said, on how common genetic variation influences human cortical development and structure. Okay, so I'm going to start with a fact that everybody knows, which is that humans have big brains. Especially humans have big cortices. So the cortex is the outer covering of the brain, shown here in cross-section. And the cortex is actually much bigger in humans relative to mouse. And specifically, the surface area of the cortex is expanded about 1,000-fold in humans relative to mouse. It's thought that the expansion of the cortex underlies a lot of our human enhanced cognitive and social abilities. Cortical differences are seen in patients with neuropsychiatric disorders versus healthy controls. We are very interested in studying what changes the structure of the human cortex. So there are some theories about what changes the structure of the human cortex. And a lot of those theories revolve around the idea that in order to create a bigger cortex, you need to produce more cells of the cortex. So the neurons, the excitatory neurons of the cortex, are produced before birth from 8 to 24 post-conception weeks during cortical neurogenesis. Almost all of the neurons are born prior to birth, but influence the size of the brain later on in adulthood. So to focus more on what the cortical wall looks like during early development, during this period of neurogenesis in fetal development, you can see a cartoon version of this here. So in the cartoon, there are different cell types. Those cell types are can be comprised into two different sections mostly. Those are the neural progenitor cells, and the neural progenitor cells can make different fate decisions. They can decide to proliferate, so they can make more neural progenitor cells. They can make a fate decision to make more neural progenitor cells, or they can differentiate into neurons. But once a neuron is formed, that neuron never divides again. That neuron is postmitotic. So if a neural progenitor cell decides to form more neural progenitor cells, which decides to form more neural progenitor cells, then you can increase the size of the progenitor pool and increase the total amount of neurons that can be produced in the cortex, thereby expanding cortical surface area. So this hypothesis was put forward back in the 1980s by Pascal Rakic, and it's called the radial unit hypothesis. And it's a way that changes in early development and cell decisions during early development can lead to changes in the size of the cortex. Okay, so that's a little bit of theory about how cellular differences can change the size of the cortex. So how do we know which genes are involved with affecting cortical structure? So the classical way that this has been done is using mouse models, where you can take a wild-type mouse or knock out a particular gene and then observe differences in the size of brain structure. In this case, a gene called EOMS, also known as TBR2, is knocked out, and you can see slight differences in the size of the brain here. In human genetics, there also has been genes that have been discovered to be involved in regulating cortical structure. Those genes, though, are mainly very rare mutations that are associated with big changes in the size of the brain. So those big changes would be microcephaly or macrocephaly. So here's an example from the homologous gene EOMS, also called TBR2, where there's a mutation about 300 kb away And this mutation is a balanced chromosomal translocation that, when homozygous, leads to complete absence of EOM's expression and a very severe microcephaly phenotype. So most of what we know about genes that affect cortical structure are either from these type of studies, very rare, highly penetrant mutations, or mouse model type of studies where we can modify the mouse, but obviously these aren't human. There's a lot of differences between the mouse and the human brain. What hasn't yet been done is to try to understand which common variants impact the structure of the human brain. So in order to identify which common variants impact the structure of the human brain, you need several things. First, you need very large cohorts, so you need a lot of people. From those people, you need to get two things. One is genotypes, and the other is phenotypes. The genotypes are generally through SNP chips and identify base pair changes in the genome. If those are single base pair changes, we call those single nucleotide polymorphisms that are polymorphic across individuals. And through MRI, we can measure the structure of the brain. Basically, we can take pictures of the brain and then measure features of brain structure. Those features can be the volume of different structures in the brain, or as I'll talk about in a second, surface area or thickness of, for example, the cortex. So in this case, you can see that individuals with the T allele tend to have a slightly bigger structure of these subcortical structures. So when we would do an association in a large number of individuals and get sufficient statistical significance, then we would say that the T allele is associated with increase of brain structure A. Okay, so now focusing on the cortex, we wanted to perform a genome-wide association study of cortical structure. In order to do that, we needed to take MRIs to get a picture of the brain. From those MRIs, they were automatically segmented. The cortex was automatically segmented using FreeSurfer, which is a segmentation tool. The automatic segmentation gives the cortical ribbon, and the cortical ribbon can be sort of phenotyped into two measures. One would be cortical surface area, and the other would be thickness, the distance from the pia to the gray-white matter boundary. So we measured two global phenotypes. One of them would be surface area across the entire cortex, and then average thickness across the entire cortex. And also, FreeSurfer allows regional segmentations. So based on the gyral and sulcal anatomy, it will divide the cortex into 34 different regional areas. From those 34 regions, we then measured average bilateral surface area and also average bilateral thickness. When performing the genome-wide association studies, we also controlled for the global measures in order to find genetic variants that were specific to a certain area, instead of finding genetic variants that were just globally impacting the brain. So in order to do a genome-wide association study, we need a very large sample size. We conducted our discovery sample in just about 34,000 individuals, and then together with another cohort, replicated in another 17,000 individuals. We did this through a consortium called the Enhancing Neuroimaging Genetics Through Meta-Analysis, or ENIGMA consortium. We founded this consortium a little over 10 years ago now to gather enough data to perform these type of studies, because each individual site is really not able to acquire these very large sample sizes to identify significant associations. So through the collaboration and sharing of many different sites, we were able to acquire a sufficient sample size to discover some genetic variants impacting the structure of the human cortex. Okay, so what did we find? So this is an ideogram showing all the different chromosomes, and then highlighting all the different loci that we were able to detect. So we detected 255 loci after replication with a nominal genome-wide significant threshold, and then 199 loci after replication with a more stringent significance threshold that corrects for all of the multiple comparisons across those 70 phenotypes that we did. So the way this ideogram is set up is that each color represents a different brain region, and the shape represents either the different phenotype or different significance threshold. So what do we notice? First, cortical structure is highly polygenic, much like almost every other trait that we know about. So there's many different variants throughout the genome which impact cortical structure. Oftentimes, a genetic variant just impacts one cortical structure without impacting many other cortical structures. So genetic variants can have specific effects on different regions of the cortex. We discovered a lot of genetic variants associated with surface area, more so than thickness. And sometimes we observe genetic variants can have an influence on multiple different cortical areas, although usually it's just one cortical area. Okay, so this shows you all of our findings plotted on the genome, but since we performed this across multiple different regions of the brain, here you can see where we found genome-wide significant effects plotted on top of the brain. So again, you can see that we found more associations for cortical surface area than we did for cortical thickness. And there is some clustering of genome-wide significant findings, specifically near the calcarine sulcus at the occipital cortex in the back of the brain. We identified the most number of associations. It's interesting to note that this is one of the early forming sulci, so it's possible that genetic variants have a stronger effect on very early forming sulci in the brain. Okay, so now I'm going to focus on some of our results for global surface area and thickness. So not talking about those regional findings right now, but talking about mainly about the global findings. So these are Manhattan plots. They represent, each dot represents an individual SNP and its association to a global surface area or global thickness. And they're plotted along each chromosome, and the y-axis indicates the significance value. So in total, we discovered 20 loci with a nominal significance threshold in the discovery sample that are related to surface area, whereas thickness we discovered much less. Okay, so now I'm going to focus and zoom in on one of the interesting loci that I introduced before. So here we thought this, I especially thought this was a very interesting locus. So this locus is near the gene eomes, and there is a genome-wide significant association to cortical surface area as shown here. So each of these dots again represent SNPs, and this dots above this line represent a genome-wide significant association to total surface area. Now, these SNPs are right near the microcephaly associated breakpoint that I talked about previously. The thought is that there are regulatory elements on this side of the microcephaly associated breakpoint that are regulating the expression of the gene eomes or TBR2. So can we layer on some more data here to understand if there are indeed regulatory elements here governing the expression of eomes or TBR2? So to do this, we're using chromatin accessibility regions. These are regions that are accessible more in progenitor cells versus those in the cortical plate than in neurons. And they seem to overlap very well with our genome-wide association locus. In addition, there is chromatin accessibility contact in the germinal zone, which contains those progenitor cells, between these regulatory elements and the promoter of this gene eomes. So this implies that these regulatory elements are actually governing the expression of eomes of this gene. So this was a very interesting locus, and in sort of sort of randomness of the universe and the combination of my postdoc work and my my start of my faculty work, we had already actually validated this enhancer regulating eomes expression. So I'll show you the results from that experimental validation. So when I was a postdoc in Dan Geschwin's lab, together with Luis de la Torre Ubieta, he did these wet lab experiments here. He designed these guide RNAs to clip out to excise that particular enhancer region. So to remove that enhancer region from human neural progenitor cells. So he designed several pairs of guide RNAs to remove that, that enhancer region, transduced some human neural progenitor cells, and found those cells that had both the left cutter and the right cutter, so the double positive cells. After that, he found that there was a decreased expression of eomes in cells that were where those enhancer elements were excised. So in total, this regulatory element is in fact impacting the expression of eomes, and both common variants, which may modify that regulatory element, and rare variants, which remove that regulatory element from governing eomes expression, both lead to effects on cortical surface area. Okay, so next what we wanted to see was if genetic variation that influences cortical structure also has effects on subcortical structures and other traits like intracranial volume and height. So first we found that cortical surface area has a very strong genetic correlation with intracranial volume or the overall size of the head. Now, because intracranial volume is also related to overall body size or height, we also performed analysis doing genetic correlation between cortical surface area and intracranial volume controlling for height. And note that there's still a very strong genetic correlation. Now, there is a genetic correlation between cortical surface area and height alone. However, there's a much stronger correlation with cortical surface area and intracranial volume controlling for height. So we do think that some of these associations that we're getting are brain specific. We do note that there is a negative genetic correlation between cortical surface area and cortical thickness. So that implies that those genetic variants that lead to increased cortical surface area may lead to decreased cortical thickness. Maybe that's, in thinking about this, we thought that this is perhaps a constraint of the skull, that those genetics that create differences in surface area may actually be constrained to fit within the skull. So you can't lead to both increases in surface area and thickness simultaneously. In terms of subcortical structures, there was a significant genetic correlation observed between hippocampus and cortical surface area, perhaps reflecting their shared developmental origins as dorsal telencephalon. But we did not notice a genetic correlation between any of the other subcortical structures and cortical surface area, implying those genetics that impact the cortex are distinct from those impacting subcortical structures. With thickness, we really didn't see a lot of strong genetic correlations with either overall head size or any of the subcortical structures. Okay, the next question we wanted to ask was which tissue types, developmental time periods, and cell types are important for governing the size of the human cortex? In order to ask this question, we used what's called a partitioned heritability approach. So we found, using existing databases, enhancers or promoters that are active within specific tissue types, developmental time periods, or cell types. We then determined if there was an enrichment of significance or heritability explained within each of these tissue-specific enhancers or promoters. So to do this, we first leveraged the Epigenomics Roadmap Consortium, which provides regulatory elements from a wide variety of tissue types across the human body. And we ran a partitioned heritability analysis and found that for total surface area, we found the strongest enrichment of heritability within brain tissue regulatory elements. Similarly, for thickness, we again found the strongest enrichment in heritability for brain tissue regulatory elements. What this implies is that genetic variation disrupts the function of regulatory elements that are active in the brain, leading to differences in brain structure. So this is perhaps an expected result, but kind of a nice true positive to see. We then wanted to pursue this a little bit further to see if we could understand developmental time periods important for regulating cortical structure. So to do this analysis, we again took the epigenomics roadmap data, but subset the brain enhancers into those that were only present in fetal brain and those that were only present in adult brain. So those are our fetal specific and adult specific active regulatory elements. We found that for global surface area, we found an enrichment in fetal-specific regulatory elements, whereas for thickness, we found an enrichment in adult-specific regulatory elements. What this implies is that for regulating surface area, fetal regulatory elements may be more important, whereas for regulating thickness, adult regulatory elements may be more important. So presumably there's certain cell types or developmental processes that are happening that are different, such that different biological processes regulate these two different phenotypes, surface area and thickness. Okay, so to pursue this one more step, we wanted to try to find which cell types are important for regulating thickness. My Roomba is going. Just give me 1 second. Sorry about this. Oh, thank you. You never know what will happen. Okay, so so next, what we tried to do was to find which cell types are important for impacting these two phenotypes, surface area and thickness. So in order to do this, we leveraged a single-cell RNA-seq dataset that was generated in the Geschwin lab, where they did drop-seq on 40,000 cells, and they found genes that were enriched in certain cell types versus other cell types. So those are cell type enriched genes. From there, we mapped regulatory elements to those. Cell types using chromatin accessibility correlation from chromatin accessibility elements that were derived from the germinal zone or the cortical plate in this time period of human mid-gestation cortex. So what this allowed us to do was to find those regulatory elements governing cell type specific genes. We then performed this partition heritability analysis again, using these regulatory elements governing cell type specific genes, and found interestingly that genetic variants impacting cortical surface area were most enriched in progenitor cells, in ventricular radioglia, outer radioglia, and different phases of the dividing progenitor cells. We also did detect some enrichment in excitatory neurons, oligodendrocyte progenitor cells, and microglia. So overall, what this implies is that progenitors cells are really important for influencing cortical surface area. And this goes back to our radial unit hypothesis. So if a genetic variant is present in a regulatory element of a neural progenitor cell, it may impact that progenitor cell's fate decisions and may influence it towards either a neurogenic fate or a more proliferative fate. So we think that the evidence we have here is human-derived evidence for the radial unit hypothesis, that genetic variation within regulatory elements impacting progenitor-associated genes prior to birth, well before birth, impact adult cortical surface area. So genetic variation at very early time period, acting at very early time periods can influence later development of the cortex and adult cortical surface area. Okay, the next thing we wanted to do was to determine if those genetics that impact cortical structure also impact any cognitive traits or neuropsychiatric disorders. So to do this, we leveraged existing genome-wide association studies from multiple different traits across many different psychiatric disorders and cognitive traits. What we found was that those genetic variants that lead to an increase in cortical surface area also lead to an increase in intelligence, at least partly, as well as educational attainment. On the opposite end, those genetic variants that create an increased risk for neuropsychiatric disorders like ADHD and major depressive disorder lead to a decreased cortical surface area. And the significant ones here are labeled. So note that we tested a lot of different neuropsychiatric disorders, and some where we know that there are differences between cases and controls in terms of surface area, like schizophrenia and autism, did not show a genetic correlation. Also for thickness, we did not see any strong genetic correlations. Nevertheless, this does imply that those genetic variants creating differences in the function of the brain also lead to differences in the structure of the brain. Okay, so far I've been focusing mainly on the global results, but we also looked at genetic variation impacting specific regions of the cortex as well. So I want to show you one hit that we had, which was extremely strong. We called it in our many Zoom meetings the monster hit. And in this hit, we have a significant locus impacting precentral surface area. So precentral surface area. This is a very strong effect for a genome-wide association study with p-values less than 10 to the negative 150. And it has a very interesting spatial effect. So the A allele affects pre and post-central surface area, decreases pre and post-central surface area, whereas the A allele is associated with increases in surface area in the parietal cortex and in the frontal cortex. So it seems like if it's increasing in one place, it's decreasing in another. And that's why we probably did not observe this for a global hit, but we did observe it for a regional hit. So this effect size was somewhat surprising for a common polygenic trait. And so I just wanted to show how big this effect size is relative to other traits. So here are some effect sizes for neuroimaging traits versus other traits. So here you can see for cortical structure, I selected the most significant associations that we detected for cortical structure, subcortical structure, which is in previous studies, and then other traits like height, schizophrenia, and educational attainment. And you can see that this particular genetic variant has a 1% variance explained effect size. It's still a small effect size for any type of association study, but it's a really big effect size for a genome-wide association study. Note that this effect size of 1% variance is much higher than was ever observed for height or diseases like schizophrenia. Okay, so another thing that we did was try to aggregate signal from genetic associations to all of the different regional surface area phenotypes, and then see if those genetic associations clustered near any known signaling pathways or biological processes through a gene ontology enrichment analysis. And what we found was a pretty surprising, very strong enrichment in Wnt receptor signaling. So those genetic variants that impact each of the different regional surface areas have effects on, or at least are enriched in Wnt receptor, enriched near Wnt receptor genes. So we thought this is really interesting because Wnt signaling is already known to impact progenitor proliferation and also areal identity in the cortex. So now this is human evidence showing that in the human, it also impacts areal identity in the cortex. And just to show one individual locus, so here's a locus that we detected as being significantly associated with lingual gyrus. And so lingual gyrus is here in the back of the brain. And the most significant variant that we detected at this locus was a synonymous variant within the Wnt2b gene, just showing one example of where genetic variants that are associated with cortical structure actually impact or at least are within Wnt genes. Okay, so I'm going to summarize this part of the talk, and then I'm going to move on to another study. So, but in summary here, we have identified common genetic variation influencing human cortical surface area and thickness, many different loci across the genome. Genetic variation influencing postnatal cortical surface area is enriched in active regulatory elements within fetal brain progenitor cells. And we believe this is human-derived evidence for the radial unit hypothesis. Both common and rare variants impacting the same gene can have similar effects, but to different degrees. And hopefully that was evidenced in the eomes story. So close regulatory elements near the gene eomes. Genetic variants altering brain structure can also alter brain function. So hopefully this was shown through the genetic correlation analysis with existing genome-wide association studies to cognitive traits or neuropsychiatric disorders. And finally, genetic variants near Wnt signaling genes influence surface area of specific cortical areas. This study was just published, and hopefully if you guys are interested in the data, you'll be able to, so you can go to our Enigma website and download the summary statistics from this data, or we have many, many supplementary tables and supplementary figures, and hopefully they'll be interesting to you as if you're interested in which genetic variants impact the human cortex. Okay, so now I'm going to switch gears a bit into a different study that we've been working on here in the lab. And this study tries to take those genome-wide association studies that we've talked about. So this would be a genome-wide association study for cortical surface area that I just described, but there's been many genome-wide association studies for a variety of very interesting neuropsychiatric disorder traits or cognitive traits. And then once we have those genome-wide association study findings, we find that most of the time, the significant associations are in non-coding regions of the genome that we don't understand very well. So those non-coding regions of the genome are generally introns or intergenic regions. What is the mechanism by which a genetic variant associated with any trait in non-coding regions leads to changes in brain structure, function, and risk for neuropsychiatric disorders? So one possible mechanism is that leading from genetic variation to risk for neuropsychiatric disorders or differences in brain traits would be that genetic variants create changes in gene regulation, which then lead to these neuropsychiatric disorders. So to explain that a little bit more, so genetic variation could impact transcription factor binding motifs, leading to differences in transcription factor binding, which then impact chromatin accessibility within a region, and finally lead to differences in gene expression. Those differences in gene expression may affect cellular behavior or circuit behavior, finally leading to altered behavior in individuals with neuropsychiatric disorders. So most previous work about genetic variation impacting gene regulation has been done through what's called EQTLs, or expression quantitative trait loci. And it's studying the link between genotype and gene expression here. There's been much fewer work studying how genetic variation impacts the regulatory elements themselves. By studying how genetic variation impacts the regulatory elements themselves, we can hope to understand two more things. One would be what's called fine mapping, identifying the causal variant within a locus. And the other would be understanding the full regulatory mechanism, understanding that this genetic variant impacts binding of a certain transcription factor, which then leads to downstream impacts on gene expression. So we're hoping that studying how genetic variation impacts this level of biology will provide new insights into explaining the mechanism of genetic variants' effect on risk for neuropsychiatric disorders. Okay, so why are we focusing on chromatin accessibility? Well, chromatin accessibility is a good indicator of the function in the non-coding genome. So most of the time, DNA is wrapped tightly around histones. But sometimes DNA is open and accessible to DNA binding proteins like transcription factors. So those open elements, those chromatin accessible elements, are usually enhancers or promoters in the genome where transcription factors or molecular machinery is able to bind to the DNA. So when performing a chromatin accessibility QTL analysis to identify which genetic variants are associated with chromatin accessibility, you need to choose a tissue type of interest. So which tissue type did we choose? So in order to address this question of which tissue type is important for many of these neuropsychiatric disorders, we performed again, a partitioned heritability enrichment analysis in the fetal cortex dissected into two regions. One is the germinal zone, which is highly enriched in progenitor cells, and the other is the cortical plate, which is enriched in neurons. And we discovered which peaks were more accessible in one tissue type than another. So those peaks more accessible in the germinal zone here, or those peaks more accessible in the neuron-enriched cortical plate here. And we found that using existing genome-wide association studies, several disorders had strong enrichments in progenitor-enriched germinal zone. This implies that this tissue type, and specifically the period of neurogenesis, is important for understanding how genetics impacts risk for these neuropsychiatric disorders. And hopefully I previously showed you that progenitors are important for understanding impacts on brain structure as well. So this implies that human neural progenitors are a good model system for studying the impacts of genetic variation on gene regulation, leading to changes in brain development and later on risk for neuropsychiatric disorders. We have been developing a large bank of primary human neural progenitor cells derived from fetal tissue. So again, when I was a postdoc in the lab of Dan Geschwin, we collected human fetal brain tissue. We sectioned off areas of the tissue that looked like cortex from 92 individual donors that were from 14 to 21 gestation weeks. We then cultured those primary human neural progenitors, cryopreserved them, and then shipped them to UNC, where I'm now a professor. In my lab at UNC, we then thawed these cells in batches and assayed two cell types, neural progenitors and differentiated neurons. So from the neural progenitors, we cultured 86 donors, and at the neural progenitor time point, they stained for markers of neural progenitors pretty uniformly. About 90% of the cells stained for either SOX2 or PAX6, which are both markers of neural progenitor cells. We then differentiated these cells for 8 weeks, and 3 weeks before we harvested them, we added a virus that specifically labels neurons. That's a AV2 virus, where EGFP is driven by a human synapsin-1 promoter. So the neurons will start fluorescing and exhibiting these standard sort of neuronal morphology. So they're immature neurons that we get in a dish, but after 8 weeks, this is about what they look like here. So then we flow sorted these GFP positive neurons and contrasted them to the progenitors and measured chromatin accessibility using a library preparation technique called ATAC-seq. So we first wanted to see if there's differences in global chromatin accessibility profiles between these two different cell types. And perhaps as expected, we saw very big differences between neurons and progenitors in terms of global chromatin accessibility profiles. Then focusing on individual promoter regions that for genes that are sort of known to be expressed in one cell type or the other, we found sort of expected patterns. So here in neurons, in synaptotagmin 13, we see higher chromatin accessibility at the promoter region of this gene in neurons versus progenitors. And for a transcription factor that's known to be involved in progenitor proliferation, we see higher chromatin accessibility in progenitors versus neurons. So this indicated to us that our results, our data makes sense, that we're getting chromatin accessibility profiles that are indicative of these cell types. But to test this a little bit further, we performed a differential chromatin accessibility analysis. So we contrasted the chromatin accessibility from progenitors versus neurons. And we found that there are many peaks where there's higher chromatin accessibility in progenitors versus neurons. For example, at the peak, at the promoter region of HOPX or Wnt2b were examples where we identified greater chromatin accessibility in progenitors versus neurons. We also found that there were certain peaks with greater accessibility in neurons versus progenitors. One that was particularly important to see was the synapsin-1 promoter. This is especially important because we use the synapsin-1 promoter to sort our cells by, so we would definitely expect that this had higher chromatin accessibility. Okay, so after we gained a knowledge of all of the different differentially accessible chromatin accessibility peaks, we wanted to compare and contrast our in vitro cultures here to our previous work using in vivo dissected tissue, which was not cell type specific, but was still in the in vivo tissue. And what we found was that peaks that were more accessible in progenitor cells in the in vitro culture were also more accessible in the germinal zone progenitor enriched in vivo tissue. Here. Whereas peaks that were more accessible in neuron in vitro cultures, those peaks were enriched in the neuron enriched cortical plate tissue. So what this indicates here is that there's some in vivo relevance to our in vitro cultures. Okay, so having satisfied ourselves that our in vitro cultures do have some in vivo relevance, we then tried to find how genetic variation in our population of progenitor cells impacts chromatin accessibility. So to do that, we determined the association between SNPs and chromatin accessibility peaks. If there's a significant relationship between a SNP and a peak, we call the SNP a CA SNP or chromatin accessibility SNP, and the peak a chromatin accessibility peak or a CA peak here. So how many of these relationships did we identify? So in progenitor cells, we had somewhat of a bigger sample size, and we identified 8,000 LD-independent chromatin accessibility QTLs regulating 4,600 peaks. In neurons, we identified 3,600 LD-independent CA QTLs regulating about 2,700 peaks. So there's many places in the genome where genetic variation impacts chromatin accessibility. So to understand a little bit more about those places in the genome, we determine if the CA SNP, the SNP impacting chromatin accessibility, falls within the peak. So this would be this color, CA SNP falls within the peak. In a lot of cases, this is what happens. So the SNP that impacts the chromatin accessibility falls within that peak, which is consistent with our idea of transcription factors binding to the peak. We also tried to determine if the CA SNP was relevant to a CA peak in other ways, including correlated CA peaks or just present in other peaks, even though they did not show differences based on genotype. And in general, most of the time, the chromatin accessibility SNP was in some kind of peak, although there are cases where we did not have a known mechanism, where the chromatin accessibility SNP was not in any peak. Okay, so our next goal was to determine if those genetic variants that impacted chromatin accessibility also had an effect on gene expression. And so to do this, we leveraged an eQTL dataset that was again generated in the Geschwin lab. This comprised fetal tissue from 235 donors, and we overlapped our dataset. So we performed a colocalization analysis to determine if CAQTLs overlap eQTLs. And we found that sometimes this happens, but not always. So for neurons, about 23% of the time, and for progenitors, about 20% of the time, does the CAQTL overlap with the eQTL. So sometimes genetic variation impacts chromatin accessibility and then leads to changes in gene expression, but not always does genetic variation impact chromatin accessibility and lead to gene expression. Sometimes genetic variation can impact chromatin accessibility, but not have observable effects on gene expression. Okay, an interesting thing for experimental design, we thought, was to try to understand if genetic variants have a stronger effect on chromatin accessibility than they do on gene expression. And so to do this, in order to avoid winner's curse, we downsampled our eQTL data to be the exact sample size of our CAQTL data. And what we found was that CAQTLs in general have a higher effect size than eQTLs for both neurons and for progenitors. We think this is interesting because it implies first that for experimental design, CAQTL studies may be higher powered than eQTL studies. Also, we think that it has important biological interpretation, which is that CAQTLs are probably impacted by a limited number of biological, there's a limited number of biological reasons that could lead to a CAQTL, mainly transcription factor binding. Whereas for an eQTL, there's multiple different mechanisms that could lead to an eQTL, including changes in transcription factor binding, methylation status, microRNAs, all these other things that affect gene expression. Okay, so one of our goals in performing a CAQTL study was to try to explain the regulatory mechanisms underlying EQTLs or genome-wide association studies. So I'm going to show one example here. This is a fetal brain EQTL zoomed in for the gene SETD9. And so you can see that there are many SNPs that are associated with SETD9 that survive our significance threshold, and they're LD buddies. But we don't know which of these LD buddies is the causal SNP, the one that's actually impacting the expression of SETD9. So we're going to try to leverage our CAQTL dataset that we just generated to understand the causal variant and the regulatory mechanism underlying this EQTL. So we can layer on our progenitor CAQTL study, and we see that there is, in fact, a CAQTL at the same location here. For that CAQTL, there's an individual SNP that impacts, that is within a regulatory element that has higher chromatin accessibility in progenitors versus neurons. In addition, there is also a neuron CAQTL observed at this locus, although to a much lesser extent, so not as significant. Okay, so now let's focus a little bit more. So we have potentially some evidence that this particular SNP is important and may be causal for regulating SETD9. So let's try to layer on some additional evidence. So first, we see that the G allele is associated with increases in expression of SETD9 in the fetal brain EQTL. The G allele is also associated with increases in chromatin accessibility. These are chromatin accessibility average plots colored by genotype. And so you can see that in progenitor cells, there's a considerably higher chromatin accessibility for the G allele. This is also observed in neurons, although to a lesser extent. So the G allele leads to increases in chromatin accessibility and increases in gene expression of SETD9. As additional evidence, we took heterozygous donors and determined if those heterozygous donors had differences in chromatin accessibility based on this same SNP. And to support our previous findings, the G allele again had higher chromatin accessibility than the A allele at this location. Okay, so this particular SNP, is it disrupting any known transcription factor binding motifs? So it turns out that this SNP is disrupting 8 different transcription factor binding motifs. So how do we know which one is important? Well, we prioritize the transcription factor binding motifs based on their expression in neuroprogenitor cells or in neurons from the single cell RNA-seq dataset that I described previously. And so in total, we can see here the REST motif. And this is the SNP, this is the location of the SNP within the REST motif. So the A allele is the motif matching allele. The G allele is the motif disrupting allele. Our proposed mechanism for this locus is that REST binds to the motif matching allele, and REST condenses chromatin, leading to a decrease in chromatin accessibility peak and decrease in gene expression of the gene SETD9. When there's a motif disrupting allele, REST is less able to bind, leading to increases in chromatin accessibility and higher expression of SETD9. So in total, we think that using these, the integration of CAQTL, EQTL data sets, we're able to go from many LD buddies to fine map causal variants of interest, and also to propose regulatory mechanisms like this one shown here. Okay, so we also, in this study, performed another partition heritability enrichment analysis to try to see if those chromatin accessibility peaks that are more accessible in progenitor cells are enriched in heritability for a variety of different neuropsychiatric disorders. And similar to our previous findings, although we see a few more diseases here, we find significant enrichments for autism, ADHD, schizophrenia, major depressive disorder, as well as intelligence, IQ, and educational attainment. For neurons, we see much less significance, although IQ and educational attainment do show significance in this as well. And then trying to tie in our previous findings from the ENIGMA study to the differential chromatin accessibility findings from this study, we did find a significant partition heritability enrichment for global surface area in progenitor and peaks that are more accessible in progenitor regions. In addition, we performed this for each of the different regional associations, and we saw significant enrichments for the colored regions. So this shows that genetic variation within regulatory elements functional in progenitors is associated with adult cortical surface area. It's interesting to see the more medial than lateral sort of enrichments. And we think that maybe our culture techniques sort of guide cells towards more medial type of neurons. Okay, and then I just have 2 more slides, and then I'll, just a few more slides, and then go to questions, so I don't want to run over. So this is just one last locus that we thought we could use. We could integrate the CAQTL and the CAQTL data with the ENIGMA data to try to understand which are important genetic variants leading to differences in human brain structure. We were particularly interested in this locus because it's at a human gained enhancer region. So a region that is present, it's an enhancer region present in human development, but not in macaque or monkeys. And this region impacts a language area. So the pars opercularis or inferior frontal region. So this locus, you can see impacts pars opercularis surface area at the HEY2 locus, and also impacts the expression of HEY2 gene. We overlay progenitor chromatin accessibility QTLs and neuron CAQTLs here, and we find that the genetic variants that are at this locus also impacts a chromatin accessibility peak right at the promoter of HEY2. And all of this lies in a human gained enhancer region. These are human gained enhancer regions here. So this is maybe a locus that has evolutionary relevance and may be important for regulating the size of a structure that's important in language. Okay, so a summary. So for this part, so genetic variation alters chromatin accessibility during human neocortical differentiation, and we were able to assay that in our human neural progenitor cell system. Cell type specific CAQTLs can lead to fine mapping and regulatory mechanisms of EQTL and GWAS loci. Overall, genetic variation has impacts during neurogenesis that can influence adult cognitive and neuropsychiatric phenotypes. And integration of QTLs across multiple biological levels allow for a mechanistic understanding of the impact of genetic variation on brain and behavior. Okay, so I just wanted to quickly say thank you to all of the many over 300 different authors that were involved in our study. So none of this would have been possible without people sharing their data. And so this giant author list sort of is a testament to the collaboration that was necessary to identify those genetic associations to the human cerebral cortex. Specifically for the ENIGMA study, I want to thank the co-leaders of the ENIGMA Genetics Working Group, Sarah Medlin, Katrina Graspy, Paul Thompson, and Neda Jahanshad. And then for the CAQTL study that was done in my lab, I want to thank Dan Liang. She's here for leading this. She's looking for a postdoc position, about to graduate soon. Hopefully her paper will get accepted, and then she'll, so if you guys are interested. And thanks very much to Dan Geschwin, where we developed all these cell lines, and Luis de la Torre Ubieta, who we developed these cell lines with, and Mike Love, a biostatistician at UNC. So I went 2 minutes over, sorry about that, Shira, but hope you guys are still there and heard that.
Thank you so much. Fascinating. We have a few questions. Jeff Waugh asks, there's tremendous variation in volume and surface area for the human visual system and V1 lateral geniculate and optical tract covary. Do you see similar functional clustering with GWAS-driven findings?
So we haven't subdivided the thalamus into different nuclei to be able to answer the difference between the subcortical structures. Is there genetic clustering between those genetic variants that impact subcortical structures and cortical structures? But if we just look at cortical structures, I don't think I actually have the slide here, but there is high genetic correlation between all of the occipital areas. So we did do a clustering analysis and find that there's a high genetic correlation between areas in the occipital cortex. Hope that answers some of the question.
Thank you. We have another question from Arthur. Wonderful work. How did you quantify differences in global chromatin accessibility from ATAC-seq data?
So for that, we used principal component analysis. So let me go there. So here, these were, so we did chromatin accessibility across the genome, and then we grouped the chromatin accessibility reads into peaks. And then from there, after there, we have a quantification of reads within peaks across all these different peaks. Then we perform principal component analysis, and then that gives us a measure of sort of global chromatin accessibility profiles.
Thank you. Jonathan Fee asks, for the first part of the talk, can you comment on what mechanistic differences might be between the growth of cortical surface area versus cortical thickness? Is there anything interesting happening between cells in 3D space, such as seeing how the axis of surface area is kind of perpendicular to the axis of thickness?
Yeah, okay, so that's interesting. So, so our thoughts about what are the differences between processes governing surface area versus thickness was that prenatal processes govern surface area, whereas postnatal processes govern thickness. We tried to demonstrate that, that progenitor cell proliferation would be an important process governing surface area. For thickness, we have less evidence as to what the biological processes are that govern thickness. Our thought was, though, that things like, that wouldn't be present in fetal development, things like synaptic pruning, myelination, things like that would lead to differences in thickness. But we don't really have a strong, like, which of those are impacting thickness at this point.
Jonathan also asks, regarding the monster hit and the mutually exclusive variation in regional cortical surface area in the precentral cortex, does surface area variation in that region affect any specific broader function, and do you feel like that hit might have larger implications?
Yeah, so unfortunately, we looked to see in all the GWAS that we could find if this hit, which is very strongly associated with surface area, has any impacts on cognition, motor movements, you know, like fine motor ability, things like that. And unfortunately, we did not find anything there. So it seems like this variant, as far as we know now, just impacts the structure of the brain without impacting the function. But it's possible that, you know, the phenotype that this does impact just hasn't been measured yet. So. But nothing now.
Thank you. So Jonathan also asks, in the second part of the talk, in the cases where genetic variation impacts chromatin accessibility, but not gene expression, does that indicate that any existing link between the initial genetic variation and the eventual phenotype, maybe risk for neuropsychiatric disease, occurs via a mechanism unrelated to gene expression? If so, what could that be?
So that's possible. Our thoughts on this were that, so a genetic variant may impact chromatin accessibility, so may impact the regulatory element, but won't lead to changes in gene expression. So what causes that? So our thoughts were that you need to have sort of some environmental stimulus, which leads to cofactors for the transcription that aren't present yet. So there has been some study about this, but in Dan Gaffney's group, showing that when an environmental stimulus is turned on, then you can observe that a CAQTL and an EQTL will overlap. In the absence of an environmental stimulus turning on, then the CAQTL and the EQTL don't necessarily overlap. So the environmental stimulus causes some other transcription factor to translocate into the nucleus. Those transcription factors together are sufficient to lead to changes in gene expression. Whereas if the environmental stimulus is not present, there's not enough change to lead to differences in gene expression. So that was kind of our thought about what about all the missing stuff here. That was our thought, is like it's environmentally mediated influences.
Thank you. Nick asks, what considerations did you make in choosing the samples to avoid issues of population stratification with, for example, clustering around specific diseases?
Okay, cool. So I think they're referring to the Enigma study. So in the Enigma study, all of the discovery sample was European. Let me go to this slide. So all the discovery sample was European based. So we had separate individuals that were non-European. We really didn't have sufficient power to do any sort of non-European meta-analysis for those. In order to avoid population stratification, we're basically trying to choose one ancestry population. We also controlled for four principal MDS components, multidimensional scaling components for ancestry to try to avoid population stratification. The lambda GCs were good, and the LDSC intercepts were all very close to 1. So all of them were uniformly less than 1.05, which all of that indicates that this avoids population stratification. So yeah, did I answer all? Was there, Shira, would you mind repeating that last one just to make sure I answered all of it?
So what issues, what considerations did you make in choosing the samples to avoid issues of population stratification with, for example, clustering around specific diseases?
Oh, okay, yeah. And so there are some about the disease question. So that sort of answers, I hope, the population stratification part. For the disease part, there are about 10% of the individuals in the study have some sort of neuropsychiatric disease, and were ascertained through a case control cohort. We did run the analysis, though, excluding any individuals with neuropsychiatric disease, and got very highly overlapping results. So we think that our results are consistent, even though we did have in this discovery sample some proportion of subjects who do have neuropsychiatric disorders.
Thank you. Arthur asks, the difference in effect sizes of GWAS SNPs on CAQTL versus EQTL is really interesting. What was the response variable in the model for ATAC-seq and RNA-seq? His concern is that the difference could be attributed to a goodness of fit due to the differences in molecular input data.
Okay, that's interesting. So we had VST normalized chromatin accessibility. So the response variable, the dependent variable, was VST normalized chromatin accessibility. And then we had independent variables, which are the SNPs. And then we controlled for PCs across genotype and PCs across chromatin accessibility matrix. For the fetal brain tissue, it was again VST normalized gene expression. And we have, and the independent variables were the SNP and PCs across the genotype matrix and PCs across the expression matrix. Yeah, but that's an interesting point. I would love to hear more about that in a non-Zoom thing. So thank you for that.
Thank you. I don't think we have any other questions at this point in time. So
okay.
Thank you so much, Jason. This was really fantastic.
Great. Thank you so much, Shira. Thanks for inviting me. I hope it was useful. Thanks a lot.
Definitely. Thanks. Bye.
Okay. Bye.